hitting the record button. Thank you very much. Okay, welcome everyone to this afternoon's workshop on an introduction to generalized linear mixed effects models. For those of you who don't know me, my name is James McBroom. I'm a statistician and mathematician within the School of Environment and Science in the new academic, or recently created new academic group known as Physics, Mathematics and Statistics. I've been an academic at Griffith for, do we still get long service medals for 25 years? Because I'm pretty much pushing 25 years at Griffith now. I came to Griffith in January of 2003. So I've been here a fairly long time. Anyway, before we crack on with the material, I'd just like to take the opportunity to acknowledge the traditional owners of the lands upon which we are meeting this afternoon, and to pay my respects to elders past, present and emerging. All right, so what we're going to do this afternoon then, as I was saying before we hit the record button, to the people in the, technology, here we go. To the people in the chat while we were waiting to start, I think I've cracked after 5 workshops the timing of these things. I don't think I've gone too nuts this time. Those of you who have been in any of my previous workshops, which was simple linear regression, multiple linear regression, generalized linear models, and 2 weeks ago, linear mixed effects models, probably remember that they all run a bit long. I will do my utmost to make that not happen this afternoon. But in any event, I'm doing my utmost to make it happen at the moment by waffling, aren't I? So we're going to introduce the idea of generalized linear mixed effects models, give you a broad overview of what they are and where they sit in the grand scheme of things. In order to understand this stuff and give you a practical guide as to how to use them, it is necessary to understand a little bit of the background theory. Now, we won't harp on about the mathematics too much, but there's some necessary maths. You don't need to understand the maths per se, but you need to appreciate that generalized linear mixed effects models are they're not a conceptual step up, but they are certainly a numerical and mathematical step up, even from linear mixed effects models that we talked about a couple of weeks ago. So there's a lot more moving parts involved in GLMMs than we've experienced before. And in order to at least appreciate what those moving parts are and to have a awareness of what to look out for when you're using these types of models, it is necessary to have a little bit of, or to talk about a little bit of the theory. So we'll recap generalized linear models, because generalized linear mixed models are built upon the GLM foundation. After that, we'll discuss what the model for the GLMM actually looks like and point out how that fits into the GLM framework that we've already seen or may have already seen before. Then we'll talk about what actually makes GLMMs so difficult to implement in practice. Now, when I say implement in practice, I don't mean necessarily from the end user's point of view. Their software exists and it's relatively straightforward to use if you're wanting to apply a GLM style model to some data, to appropriate data. What I mean is why the software is so specific and specialized, I suppose. So from my point of view, as a statistician who implements these sorts of algorithms or researches the best types of algorithms for these various models, the most numerically efficient and so on, why they are so interesting and potentially difficult, and what repercussions that has for you as the end user. Or maybe some of you aren't end users. Maybe some of you are genuinely interested in the nuts and bolts of these things as well. What makes these things so potentially tricky? So we talk about consequences of the approximations we need to make in order to actually get some numerical estimates based around these models. Then hopefully that I won't drag the theoretical bit out for too long. And that will leave us then to talk about some practical aspects of implementing GLMMs on data analysis. In particular, what are the various estimation methods available to you and in which software they exist, or which software uses which type of estimation method. I will have a very R-centric view this afternoon, but there are some notable other pieces of software, for example, SAS, Stata, some Bayesian bits of software we'll make a little mention of here, but I'll be focusing on the R stuff mostly this afternoon. So when we talk about this bit, it'll be mostly about R. Then we'll move on to things to consider when you are postulating your initial plausible model. Looking at the data, considering how the data was collected, and the various components of it after you visualized it, for example, and so on, and postulating the initial plausible model that you might fit before you then go on to refine that model, check it for diagnostic issues, assumptions, and that sort of thing. Okay, so how we go about that, because there are a lot more moving parts now in generalized linear mixed effects models, as I mentioned. Then we'll talk about fitting and selection. So how we just determine, for example, which of a group of covariates we should include in the model, which ones we may be able to safely remove from a model, the random effects variance covariance structure that we may specify initially may be reasonably complex. Can we reduce that and make it simpler in the interests of parsimony? Doing that sort of model selection depends very much in generalized linear mixed effects models upon the estimation method that you have used with the software you selected. And we'll discuss that as well, because that's important. Assumption checking, there are some nice packages available to us now, in R in particular, that allow us or take into account the fact that we don't have normally distributed residual or response data anymore. We're in a generalized linear model world now where the response could be dichotomous. It could be counts. It could be over dispersed counts. It could be counts with too many zeros to be accounted for by standard counting models, et cetera, we've got some nice assumption checking software, visual stuff, and hypothesis-based stuff, which I'm not as big a fan of, but we'll talk about that as well. How we then go about interpreting the estimates from the final model after we've gone through the model fitting, selection, diagnostic, and refitting, reselecting, re-diagnosing process. Once we get to the final model we're interested in, how do we then interpret those estimates? Then, time permitting, and time will permit, I guarantee it, we'll have a look at a couple of examples. So briefly, what are generalized linear mixed effects models? Well, you can think of it in two non-exclusive ways, actually. One way or the other way, they don't exclude each other. One way is you can think of generalized linear mixed effects models as a class of model that extends generalized linear models to include random effects. Because, for example, you've collected binomial or binary data or plus-on count data through time, repeatedly measured or in groups. So for those people who came to the linear mixed effects modeling workshop a couple of weeks ago, we spent a fair bit of time, I'm not going to go over that again today, but we did spend a fair bit of time in that workshop discussing the nature of grouped data and why it creates problems like within group dependency and between group variation that we need to account for. Generalized linear mixed effects models allow us to do a similar thing in the generalized linear model situation that we use linear mixed effects models to do in the linear model situation. So it's analogous, parallel stream. The other way you can think about generalized linear mixed effects models is a suite of models that extends the linear mixed effects model to situations where we've got certain types of non-normally distributed response variables. So not every non-normal response variable fits into the GLM or GLMM model class, but most of the interesting and useful ones do. Things like, as I've mentioned before, the binary, the binomial plus-on, negative binomial, zero inflated models, zero inflated plus-ons, hurdle models. They all can potentially fit into this framework. So GLMMs are possibly the most flexible framework of, or they're not possibly, they are the most flexible framework of model that we've dealt with in at least my set of workshops for RED last year and this year. So for us, in a way, GLMMs are the culmination of the linear model work, the generalized linear model work, and the linear mixed effects stuff last week. So GLMMs are the hybrid offspring of all of those things put together. And so they're necessarily potentially complex. Data grouped, we've talked about that, repeatedly measured. There can be a hierarchy of groups. For example, some of us in here would be aware of student evaluations of teaching or courses. So we could be doing a study to evaluate evaluations, maybe as part of the AECD process or whatever that just recently happened, evaluating people's SETs or SECs within academics. Did I just hit the button? Went back. Within academics, most academics teach into more than one course and therefore will have multiple SETs or SECs associated with them. Within their academic discipline, within various schools, within the groups, within various universities. So you can start to build hierarchies here or nested hierarchical models that span many, many hierarchies. We can also have crossed random effects as well. We talked about that in the linear mixed effects case. And this is just a slide from the linear mixed effects models that we looked at a couple of weeks ago. So there's two main issues when you collect data in groups, when you repeatedly measure things. That is, within each of the groups or clusters, there's generally a lack of independence because you're repeatedly measuring the same thing, and there are things intrinsic to that person or group or university or school within a university that mean that observations taken on them repeatedly will tend to have some similarity. Okay. We assume, though, that between the groups are independent, so the various groups are independent of each other. But although we can not limit ourselves to just that assumption, but generally we assume the groups are independent and the observations within the groups will have some dependency. There's also increased complexity in the variability structure that we observe when we have clustered data, because now not only do we need to care about the sort of observational level random effect or the observational level variation, which we call the within group variation in these situations, but now we also need to try to account for the idea that groups will vary from one another as well, and we need to quantify that variation between the groups as well. So, and obviously, as you get more and more levels in a hierarchy, for example, in a nested design, like we talked about before, then you'll have, for example, you'll get between university variations in the SECs, you'll get between groups within various universities, variations in SECs, you'll get between schools within a groups, variability between SECs, you'll get variation between academics within a schools, SECs, and so on. All of these things need to be accounted for in a hierarchical structure. So this stuff can get complicated if you have collected data in those sorts of complicated ways. We did discuss nested and cross designs a fortnight ago. I'm not going to go back over this in any sort of detail, except to say that nesting is reasonably common. right? Certainly when you've conducted a hierarchical type study, like I just mentioned with the universities and the groups and the schools, etc. And a nested design, subgroups are unique to their higher level group. Okay, so as an example, teacher within school, as long as the teacher doesn't work at two separate schools, sites within catchment, and so on, right? So that would most likely be nested. You just need to look at the groupings within a broader group and make sure that they are unique to that broader group, and you have, in that sense, nesting. The alternative to nesting, and we're creating a dichotomy here really with the definition I've put, is crossed. But in truth, you can have mixes of crossed within hierarchical models and so on and so forth if you needed to. But generally speaking, if there's any crossing whatsoever, even if it's within a hierarchy, we tend to call it crossed, because crossing creates extra issues algorithmically and from an interpretability point of view that we need to think about more carefully. So just to mention to you guys that for what we're doing this afternoon, I'm just going to be looking at a single level, a single layer of nesting. We won't be looking at cross designs. If you suspect or know that you have data that are crossed, I'd be more than happy for you to reach out, email or whatever, and arrange a time to discuss. I'm off overseas on Monday for a week. And then, of course, once we come back, it's all guns blazing for T1. So if you do email me, please be a little bit patient. Besides all the teaching that's beginning and the T3 courses that have to be wrapped up and going away overseas for a week, I'm also trying to get the statistical consulting unit up and running as well. So I've got a lot on my plate at the moment. So it may be a week or so before I get back to you if you do send me an email, but I will get back to you. Okay, so definition of cross I've put here, just not nested. And with crossed, some or all of the subgroups belong to some or all of the higher level groups. So for example, I'm a perfect example of this. I teach mathematics courses into two schools. I teach a couple of first-year math courses to both students within the School of Science and students within the School of Engineering. Some of my physics colleagues do the same for the physics courses. I even got help those poor students last year. I even convened a course called Aviation Science last year for the aviation school. Well, actually, aviation, I think, is a sub-discipline of the engineering school. But you get my point. If we were looking at the SECs for my teaching, and we were looking at those maths courses that I teach into two separate schools, then I would be crossed because I am not unique to the one school. Okay, so that's an example there. All right, now let's look at an example data set. So by the way, guys, please stop me if anything's not clear as we go through, but hopefully so far I haven't said anything too momentous. I've just hinted at things momentous to come, perhaps, potentially momentous, I'm not sure. So let's look at an example data set that's fairly typical of a basic form of data that you might get that's collected. Well, this is an example that I worked on when I was my first job out of uni, actually, when I was in Darwin, working as a biostatistician for a year before I went overseas to do my DPhil. This was an in situ clinical trial in an Indigenous community on Bathurst Island, which is in the Gulf out of Darwin, on Tiwi people. It was on children, and bacterial infection in their nasopharynx. We noticed that these Aboriginal children were almost continually infected with bacteria, and so we had a clinical trial to see if a certain drug was beneficial to trying to get rid of that bacterial load. So we followed numerous children through time, which is the week here. So we followed them as 0, 2, 4, 6, and 11 weeks, with some missing here and there. So each of these children were repeatedly measured. Half the children were randomly allocated a drug, the other half were randomly allocated a placebo. And it was my job as a statistician every 6 months to, for ethical reasons, as part of the ethics approval, to take a dip into the data to see whether the treatment was working or not. And if it was working, I had the responsibility of calling off the trial, which was an interesting job for a 22-year-old straight out of uni. But anyway, we won't talk about the academic pressures that come to bear in situations like that when there's 5 years worth of funding involved. However, so this is typical. This is typical of the sort of data that we might get repeated measures in something like a clinical trial, following people through time. So by the way, sorry, the response here is the column called Y, and you'll see it's Y and N, yes and no, presence or absence of bacteria when they analysed the swabs from their nose, essentially. AP is active or placebo, so that's the drug. High-low, because it was a remote Indigenous community, there was some issues with compliance to the regime. So it was a blind study. In fact, it was a double-blind study. In fact, I think I was the only one who knew who was in which group for this study. But there was an issue with some compliance to taking the drug every day. And so there was a high and low compliance group as well, randomly allocated within each of the placebo and active drug groups. High basically meant that the community nurse would pop by and remind people you need to take your tablets. And low was people were left to their own devices to remember to take the tablets or not. Anyway, the reason I'm explaining this data in detail now, rather than just skimming over it, is that this will be one of the examples we look at analysing toward the end, at the end of the workshop. All right, so let's get into the fun stuff, which I know you're all begging to have, which is the mathematics. So let's quickly recap generalised linear models. This is from last year's workshop. Maybe it's just a recap of stuff you already know. I'm going to go through it fairly quickly. If there's anything that is causing you major grief, please stop me. But you can take my word for all of this. Trust me, I'm a statistician and I wouldn't tell you the wrong stuff if you prefer to just sort of take it on faith. All right, so there are several components to a generalised linear model. There's the response that we assume is a random sample. It's independently drawn from a population, and each of those data points drawn from that population have an identical distribution. We give that distribution a generic mathematical name, if you like. We call it FY. Okay, so our data are drawn from a distribution. Now, in generalised linear, a population with a distribution. In generalised linear models, these distributions come from a one parameter, or assumed to be from a one parameter exponential family. Now, that's a very general class of probability distribution family, and it includes things like the binary distribution, or Bernoulli, the binomial distribution, which is aggregated independent Bernoullis, the Poisson, the negative binomial, the multinomial distribution for multiple categories, gamma distribution. The normal distribution is also a member of the one parameter exponential family. So you can see that the word general here in GLMs lives up to its name. It covers a lot of really important situations that we encounter in everyday data collection situations. Okay, the response has a mean. Now, the form of that mean will vary depending on what the distribution is that you're talking about. So, for example, with a Bernoulli random variable, the mean is the probability of success, which we denote P. In a binomial generalised linear model, or a binomial distribution, the mean of a binomial where there are n trials and probability of success is the p, the mean is n times p. For a Poisson with a rate lambda, the mean of the Poisson is lambda. Okay, so this mean mu here is just a generic expression for the mean, and it depends on whatever distribution you're talking about. The variance is an interesting thing in generalized linear models because in all of them except the normal, the mean will, the variance of the random variable will depend upon the mean. And that dependency, the structure of that dependency, how the variance changes as the mean of the distribution changes, is captured in this function that we call v bracket mu. So v bracket mu is known as the variance function. Okay, and it's a mathematical description of how the variance varies or changes with the mean. I won't bore you. There's a few examples on the next slide. I won't walk you through any of them now, or will I? I'll just do a quick one. So for a binomial, okay, so for a binomial with number of trials n, probability of success p, the expected value or the mean of y is n times p, as we just talked about, and the variance is n times p times 1 minus p. Okay, so there's the mean. There's the mean repeated. There is a relationship between the variance and the mean. The variance will change as the mean changes in the binomial. And that's true in the Poisson, for example, with rate lambda, the mean is lambda, and the variance is also lambda. It's the only statistical distribution that has that property, in fact, unless you make up some sort of, synthetic example. But yeah, so the variance depends on the mean in generalized linear models, and therefore also in generalized linear mixed effects models. Okay, the other component to the variance, there's a symbol called phi. Now that's known as the dispersion parameter. For the binomial, Bernoulli and the Poisson, the dispersion parameter is, it's a mathematic, you can show this mathematically, we won't do it, of course. But for the Bernoulli, binomial, and Poisson, the dispersion parameter is 1. Theoretically speaking, it is 1. But in other distributions, for example, the normal distribution, the dispersion parameter is sigma squared, the variance that we know and love from, basic stats. Okay, and in other situations, the dispersion parameter takes other forms. But the important bit here is that the dispersion parameter is written on its own outside of the variance function because the dispersion parameter, phi, doesn't depend on the mean, but it still contributes to the variance of the response variable. Okay, so mathematically we need to write them separately, and it's convenient to do so as well for various reasons that we don't need to go into. Okay, now the other part you may remember, if you've worked in these models before, you'll know this. If you did the workshop with me last year, you may remember that another important component of the generalized linear model is what's called the link function. We call that function G. It's a function of the mean, and it helps us relate the mean to the linear predictor. So the linear predictor is the beta naught plus beta 1 x1 plus beta 2 x2 part of the model, the bit with the explanatory variables in it. We call that the linear predictor. Part of the role of the link function is to is as a way to convert a linear predictor, well, the inverse link function, I should say, is a way to convert the linear predictor back onto the scale of the mean, whatever that might be. So for example, the mean in a Bernoulli is a probability. So the mean lies between 0 and 1, right? Because probabilities lie between 0 and 1. But if we can't model something that lies between 0 and 1 by covariates that can take any value we like. So we need a way to, let's just say, compare apples to apples. And that's partly the role of the link function. Now, you may or may not remember, but in the workshop we did last year, we assumed, or we focused mostly on what's known as the natural or canonical link functions. These are the link functions that fall out when you do the mathematics on a one parameter exponential family. So for the binomial, it's known as the logit link function. For Poisson, the canonical or natural link is the log link. For gamma, it's the inverse. For the normal, it's the identity, etc., etc. Did someone just type something in chat, or is that just my... Nope. Okay. All right. So each distribution in the generalized linear model family has a unique natural link, and we focus mostly on that. But other links do exist. For example, in the binomial case, there's something known as the complementary log-log link. For instance, there's also, economists have used what's known as probit links for a long time, which is the inverse of the cumulative distribution function of a normal distribution, standard normal, and so on and so forth. Lots of functions can perform the functional role of a link function, but only the canonical links have certain nice statistical properties. And just to keep things simple, we're just going to focus on the canonical links. Doesn't mean you're limited to them, just means that I don't have enough time to talk about all the rest of the stuff that goes along with not using them. Okay, our linear predictor, as I said, is that linear combination of the betas and the x's for the covariates and explanatory variables, the things we think may have an influence on the response. All right, so a couple of examples. We've already done the binomial or the Bernoulli. Okay, so the mean is P, the variance is P1 minus P. So, and the dispersion parameter is 1 for Bernoulli, which is a binomial with a single trial. For the Poisson with rate lambda, the variance equals the mean and the dispersion parameter is 1. In a normal distribution, the variance function is 1 because in the normal distribution, the variance doesn't depend upon the mean. That's why in linear regression models where we assume normally distributed error terms, that's why we're looking for constant variance, okay? Because we don't expect in a normal distribution the variance will change with the mean. Okay, so they're just a few examples. All right, and this is a table that you may have seen before as well if you did the GLM workshop with me last year. But here's a few of the common, not exhaustive list at all, but a few of the more common distributions in the one parameter exponential family or belonging to the GLM class of families. Binomial, Poisson, gamma, and normal, and their canonical links. So we've got the logit, which is log of the odds ratio. We spent a bit of time talking about that, the GLM workshop. The Poisson, the log of the mean. The gamma is the inverse. The normal is known as the identity. We don't do anything to the mean in the normal distribution. Okay, and there are others as well. There's negative binomials, which use a log. There's multinomials, which use a form of logits as well, and so on. All right, so now we all thoroughly understand GLMs, right? Big thumbs up in chat if you no, like don't you don't need to do that. Now that we thoroughly understand GLMs, let's extend that idea to generalized linear mixed effects models. We have a response. Now we need more than one subscript now, because now what we're doing, we're keeping it as simple as possible, which is one level of nesting here. So repeatedly measuring observations on numerous groups of data. We need a subscript that keeps track of what group we're in, i.e. what infant we are measuring when we're measuring their bacterial carriage in their nasopharynx. But then we also need another subscript to keep track of which of the observations we're talking about on that particular infant. So we need a subscript, which we normally use J, or the next one in the alphabet, to keep track of which groups we're talking about, and I keeps track of which observation within each group we're talking about. So we're going to say our response is denoted by Yij, where J goes from 1 to Ni. So we're allowing each of the I subgroups, or each of the M subgroups, I should say, to potentially have different numbers of observations. You may have recalled the example data set I showed you. Some of those children were measured 5 times, some were measured 6, some were only measured twice. Okay, so we need to be flexible in our nomenclature. We have what's known as a conditional distribution. So given the random effects, given that we are going to allow each of these groups to randomly differ from each other, the distribution of the response variable is independent and identically distributed with a certain distribution. Now, this distribution F will be one of our generalized linear model distributions: a binomial, a Poisson, a negative binomial, a multinomial, a whatever it might be. Okay, we still have a link function. That hasn't changed. Okay, so we're still relating some function of the mean of that conditional distribution to the linear predictor of covariance. That's exactly the same as in a generalized linear model. Now, what's different, however, you may be guessing what's coming, is that into the linear predictor now, to account for the idea that we could have correlated observations within each group, and we also need to account for the variation between the groups, the extra variation that arises because we've collected data in this sort of grouped fashion. We now not only have the fixed effects component, which corresponds to the generalized linear model, but we add random effects into the linear predictor. Now, these random effects are U0, U1, etc. So U0's a random intercept, U1 is a random slope associated with the first X variable, etc., etc. Okay, we add those random effects in, and they basically perform the role of inducing a correlation into the observations within each group, and they also allow us to obtain estimates of the between group variation. And it's just a matter, in this case, of determining how complicated we want our random effects structure to be. To be perfectly honest with you, in most generalized linear mixed effects models, most people go simpler, go for the simplest model, because GLMMs are numerically difficult to control, and we'll talk about why a little bit later in the talk. Okay, so generally speaking, we don't have a million random effects like I've drawn here. This is just for generalization, this particular model. Normally, it'd be very rare in most situations to have more than 2, maybe 3 at tops random effects per cluster in a GLMM. I mean, there are always situations where those sorts of rules of thumb don't apply, but in many, many situations, you only need 1, maybe 2 or 3 at the top random effects. Okay, so a random intercept and maybe 1 or 2 random slopes, depending. All right. Now, we assume that these random effects, however many we have, let's just suppose we've got Q plus 1 random effects. So Q slopes and 1 intercept. So Q plus 1 random effects. We assume that they are independent, identically distributed, drawn from a multivariate normal distribution of dimension Q plus 1 with a 0 mean vector and a Q plus 1 by Q plus 1 square symmetric positive definite variance covariance matrix. We spent a fair bit of time 2 weeks ago talking about variance covariance matrices. We're not going to really go on about them again today. All right. All right. Now, this is an even maybe more broad definition of GLMMs that also basically covers linear mixed effects models as well. And that is that, but it basically, is what I've just told you. We've got a response variable. It has a conditional distribution. The mean of that conditional distribution is the inverse of the link function. And it also depends on variance function, scale parameter, and so on. The linear predictor is a combination of fixed or deterministic components and stochastic or random effect components. Okay, so this ZU bit here is actually, the ZU bit here is actually this random effects component, just written in a different way. Okay, where the conditional modes, what we call the random effects, are the conditional modes of each group, have a multivariate normal distribution with a zero mean vector and a variance covariance matrix that depends on parameters theta. And those parameters theta need to be estimated. Generally speaking, they're the tricky bits in generalized linear mixed models. They're the ones that cause the problems, which is why in practice, people don't go for very complicated random effects structures, because the more complicated you get with your random effects structure, the larger your random effects variance covariance matrix needs to be. And unless you want to make some very restrictive assumptions on the form of that variance covariance matrix, for instance, that there's no correlation, that it's got equal variance, so the main diagonals are the same value, so you only got one parameter estimate, for instance, which is a very restrictive assumption to put on. Unless you're prepared to put on really restrictive assumptions on that matrix and the parameters in it, you're going to have a lot of parameters to estimate. And probably now's not the time to mention this, but I'll mention anyway. The method of choice in these models for estimation, which is known as maximum likelihood estimation, struggles traditionally with giving you decent estimates for variances. So when you combine a complicated random effect structure with an estimation procedure that struggles to give you good estimates of those parameters, making things massive and complicated at this point is sort of a recipe setting yourself up to fail a little bit. So, but anyway, we'll talk about some practical strategies of how to handle that potential. It's not really a paradox; it's just an issue with these models as we go through. All right, now here's where, and I do apologise for this, but I have this integral burnt onto my brain. I spent 4 years of my life during my DPhil in the UK studying this thing. The GLMM, the function that we need to optimize over in order to get estimates for the betas, for the fixed effects and the random effects variance covariance matrix parameters, involves an integral. It looks a little bit like this. And unfortunately, in all cases of exponential family distributions, except the normal distribution, you can't solve this integral analytically. There's no closed form solution to this integral. You need to approximate this integral somehow in order to perform inference in generalized linear mixed effects models. Now, this is where generalized linear mixed effects models and linear mixed effects models part ways in terms of straightforwardness. Even though they are deceptively similar in what they're trying to do, they are technically very, very different to each other in terms of numerical procedures and algorithms and the potential for things to go badly wrong. If you don't know what you're doing when you're coding algorithms or even creating algorithms. Okay, this bit here, I don't know if I can highlight it, but this bit here, okay, that bit here is basically the probability distribution function of a multivariate normal distribution. So it's the distribution of the random effects that we assume. This sigma here is the covariance matrix of the random effects. This thing here, Fy given u, that's the conditional distribution of the response given the random effects in the model. And it's one of these exponential family distributions like the binomial or the Poisson or the multinomial or whatever. So when you put those two things together inside an integral, it's slightly it's not chaotic. I shouldn't say it's chaotic. We just can't, we have no closed form solution to this integral. Okay, so if we can't solve the integral, we can't get the likelihood. If we can't get the likelihood, we can't find the parameters that maximize that likelihood, which is basically statistical parameter estimation 101. It's maybe not, it's kind of the gold standard of parameter estimation techniques in classical statistics. Of course, you don't have any of these sorts of concerns if you're doing Bayesian work. And in many ways, Bayesian is a lot more flexible and robust, perhaps, in these sorts of models than frequentist. However, the downside of Bayesian is that there's a large cognitive load associated with getting your head around Bayesian to be able to do it properly, rather than just, you know, copy code off of Google and hope. You need to understand probability distributions. You need to understand how to manipulate them. You need to understand how to form conditional models and so on. There's a few more moving parts with the whole Bayesian process. Plus, it can be potentially slow, and there are issues with the algorithmic approach to drawing samples from the posterior distributions in Bayesian analysis involved with Markov chain Monte Carlo or various other Monte Carlo techniques you might use in those situations, like the idea that Markov chain convergence to the equilibrium distribution is only guaranteed asymptotically. So, you know, how long do you run these chains? How many parallel chains might you run? There's all sorts of things extra complications that arise in Bayes. So we're not going to talk about Bayes this afternoon. But again, if you're interested and are working in that area, I'm more than happy to have a chat with you if you wish. Okay. All right. So that's the integral in the room. It's that thing there. It is a bit of an elephant in a way. And like I said, this thing dominated my life for about 4 years, 25 years ago now. As I mentioned, those integrals are analytically intractable. We can't find a closed form solution unless Fy given u is a normal distribution, in which case we've got the linear mixed effects case. Please go back and see the workshop that I did on that 2 weeks ago. So therefore, in order to be able to use the maximum likelihood technique to get estimates for the parameters, we need to approximate this integral somehow, or this expression somehow. And there's two ways we do that. One is we muck around with the integrand. Now, the integrand is the bit inside the integral that we want to find the antiderivative of, if you recall any of your maths. So the integrand is the nasty expression involving Fy given u and the normal multivariate normal distribution stuff here. So this bit here is the integrand between the integral sign and the du sign. All right. So one way to go about this is to do clever things to the integrand, in particular to FYU, but the whole thing basically, to make the integrand look more like a multivariate normal distribution, especially at important parts of the integrand, Taylor series expansions and stuff like that, if that means anything to anyone. Again, not our job this afternoon to go into the nitty-gritties of the numerics of this. But so one way is to muck around with the integrand, do things to it to make it look like an integral that we can solve. Okay. The other way is to just go straight ahead and approximate the integral using a generic term for numerical integration, that we call quadrature or cubature, depending on which part of the mathematical world you live in. Okay, just if anyone's curious, and I have no reason to think you would be, but I'll say it anyway. I focused mostly on the numerical approximation to the integral approach. And in fact, I'm going to have a small flex here now. I wrote the first algorithm and package in R that does what's called adaptive Gauss-Hermite quadrature for generalized linear mixed effects models. I wrote that back in 2000. Never published it on CRAN or anything, but if you're ever interested, I still have the package. It works very well. I use it a lot. I can flick you a pre-compiled version if you want to take a run at it. But there are plenty of other good options for software that have developed in the last 25 odd years. So I won't be heartbroken if no one contacts me to get a copy of my package. Okay. All right. So what are the consequences of these two approaches to approximating this likelihood? There's always consequences when we approximate things. There's always trade-offs. Don't get something for nothing. So approximating the integrand, which I call analytical approximation. There are two main approaches that you'll see in the statistical literature that would fall into this category. One's called penalized quasi-likelihood. The other one's called the Laplace integral approximation. Now, the Laplace integral approximation it kind of has a foot in each of the camps, actually. You can view it as an analytical approximation because it does clever things to your integrand in order to make the integral work for you. But it is actually also a special case of very simple Gaussian quadrature as well. So it's also the simplest sort of numerical approach to numerically approximating the integral. But these analytical approximations, fiddling around with the integrand, are generally quick, but can be dirty, can be quite rough and ready, especially PQL. They're quite easy to implement. If anyone's ever used the GLMM PQL package in the maths library in R, I developed the algorithm for that and gave it to Brian Ripley. Brian Ripley was my DPhil supervisor at the time. And Brian coded that up and put it into maths. It's very simple. If anyone's interested, it's analogous to how we linearize the response in a GLM and then use linear model via a Taylor series expansion, second order one, and run a linear model on that linearized response. It's analogous to that. Okay, which is why you can use existing LME software. You linearize the response variable in the GLMM, and then you use LME software, essentially. They suffer from bias, especially when the approximation is poor. Now, when's the approximation poor? We'll talk about that, but the approximation is poor when the means are low. So if you've got binary data, for example, or you've got plus-on count data with a small mean, low counts, for example, or also they tend to be poor when the variance components of your groups of your random effects are large as well. Okay, so these are now the cons, right? So the pros are they're quick and easy to implement. The cons are they suffer from bias. You have no control over the approximation error. Now, you might be like, I don't quite understand what you're saying there. Well, compared to numerical quadrature, compared to numerically evaluating that integral using numerical techniques, you can't do anything about the approximation error in these. It is just what it is. It's an analytical approximation. Whereas in the numerical integration techniques, if you suspect that you are not estimating that integral very accurately, you can always increase the number of Monte Carlo points that you use or the number of numerical quadrature points that you use. You might be using 5. You're not quite sure about the approximation. You could up it to 15, for instance. So there's always something you can do. You can always increase the number of points in the numerical approach, but in this analytical approach, you're stuck with it. Okay. Another downside is the penalised quasi-likelihood, not Laplace. This doesn't apply to Laplace, but to the penalised quasi-likelihood one, it doesn't yield usable likelihood metrics. So you can't get, or you can't use PQL estimates to do likelihood ratio testing for model comparison, for instance, or model selection, AIC, and so on. Laplace is okay because it is, as I said, it's got a foot in the other camp as well, but PQL doesn't yield those things. And you'll notice, actually, if you use penalised quasi-likelihood software in R, or hopefully other packages that are responsible in how they've coded it, we made sure, for example, that GLMM PQL in the mass library puts a missing value for the AIC, the BIC, the likelihood, and so on, because we didn't want people to be using it. Okay, now what about the numerical, approximating the integral by numerical approximations? So this is Gauss-Hermite quadrature is the method of choice here, although you could use any integration technique you wanted, but Gauss-Hermite is tailor-made for this sort of stuff. We approximate that integral essentially by a weighted sum, where the weights and the points at which we evaluate the integrand are carefully and cleverly chosen. In the case of Gauss-Hermite quadrature, those points are chosen as the roots of the Hermitian emission. Sorry, my French is poor. Polynomials. Okay. Also, you can use what's known as adaptive Gaussian quadrature. So I mentioned that my package was the first one to use adaptive Gaussian quadrature. There was another package at the time, it still exists today, called GLMMs in R. At the time, back in the early 2000s, it was just a straightforward non-adaptive quadrature, which suffered accordingly, but the author of that, Gore and Brostrom, has since made it adaptive. However, adaptive means that you recenter and rescale the integrand through each iteration of the process. So as you get estimates for your conditional modes for each group, you move your quadrature points so that they are centered and scaled around the central part of that conditional mode, if you like. You center the integrand around the conditional mode and do the integration on that. And that puts more of the points in more of the more important parts of the integrand, which means it's slightly more efficient and quicker. Okay. You can also do Monte Carlo integration, Markov chain Monte Carlo, Bayesian context, or frequentist. Doesn't really matter. That's not as big an option. You don't see Monte Carlo used a ton in frequentist, but you do see Monte Carlo in the form of Markov chain Monte Carlo used almost exclusively in Bayesian work here. Now, these techniques are generally slower but less dirty than their analytical approximation counterpart. They're definitely more difficult to implement in software, a lot more numerical considerations to think about. And probably their biggest drawback is it's only really feasible to do this sort of stuff due to the nature of how these numerical integral approximations work when you've got multiple integrals. It sort of gets geometrically large problem very quickly. Only feasible for small to moderate dimensions of random effects per group. And in fact, in putting this talk together, so when I wrote my software back in 2000, I made it available for up to 2 correlated random effects, because that was all I could manage at the time with the computing power we had. I had expected in the intervening 25 years that some of the adaptive Gauss-Hermite quadrature approaches would have maybe extended that to 2 or 3 or maybe even 4 nested random effects. Well, turns out that at least of the ones I've looked at, they're only doing random intercept for adaptive Gauss-Hermite quadrature at the moment. If you want more than just a random intercept per group, you need to look at Laplace or penalized quasi-likelihood or some other method right now in R anyway. Okay. An advantage of these ones is you do have control over the approximation error, so you can increase the Monte Carlo sample size or the number of quadrature points if you suspect your approximation isn't particularly accurate. And probably the biggest bonus or pro to these is they do yield usable likelihood metrics. So you will get likelihoods out that you can use to perform likelihood ratio testing or use to construct a Kicky's information criterion and so on. Just as a note before we move on, adaptive Gauss-Hermite quadrature with 1 point is mathematically equivalent to the Laplace integral approximation that we talked about on the previous slide. And at the moment, at this point in time, this has been an ongoing issue in GLMMs for a long time. This may not mean anything to you guys, but if you do remember us, the talk I gave, the workshop I gave 2 weeks ago on linear mixed effects, I spent a little bit of time talking about something called the residual or restricted maximum likelihood estimates, REML. At this point in time, it's still an open question in statistical research. There have been some methods or equivalent REML methods postulated, but there's no definitive REML available to you in generalized linear mixed effects models. So none of these things account for the fact that you're going to get some bias in the random effects variance estimates due to the fact you haven't adjusted for the estimation of the means in the fixed effects. So there's no easy and simple and obvious REML equivalent in GLMMs as yet. Okay, now this would be a good point. Do you guys want to just keep smashing through, or would you like 5 minutes to have a short break, collect your thoughts, and then we'll plow through the practical stuff? The practical stuff will go a bit quicker, and then we'll get on to at least one of the 2 examples. Any thoughts? Do you want a break? It's been a long hour. I'll make the decision for you. Let's take a short. Oh, you want to keep going? Quick break. No, yeah, look, let's take a quick break, guys. I reckon 5 minutes max. Okay. Let's meet back here at about 12 minutes past 2. I've got 7 minutes past on my clock. Okay. So go grab a quick cuppa or whatever, stretch your legs, and I'll see you back here in a minute. Okay, I hope people have managed to grab a quick break. We're back. Okay, let's keep going. Oh, good, good. Okay. All right. So that ends the theoretical stuff. So you can all breathe a sigh of relief. The maths is basically over. Okay. And now we're going to look at some practical things. Now, this will just be a set of guidance and guideline tips, if you like, to help you with the analytical process using generalized linear mixed models. Right, so I'm going to sort of run through these. I've made these slides available fairly quickly. I've made these slides available to you. Hopefully you've all been able to access them in the chat. I think I put them up yesterday. I'm hoping the best way to show people how to do this stuff is via examples. So I'm kind of hoping to be able to leave myself half hour or so, which only leaves about 17 minutes for me to get through this. So enough prevaricating. Let's crack on. Now, I based this table that you're looking at now fairly heavily around Ben Boker's GLMM FAQ webpage. It's a useful place to go if you're working with GLMMs. If you just Google GLMM FAQ, you most likely will find it in the first couple of results. Once you look past the very helpful AI search assistance that you get nowadays in your browsers. So I've added a few things and taken a few things away that I don't necessarily agree with him on out of this table. But essentially, if you're looking for PQL, which given that Laplace is so ubiquitous now, the Laplace approach is so ubiquitous, I think PQL approaches are a historical legacy that I wouldn't necessarily recommend you try unless everything else fails. All right, so if you have a choice between PQL and Laplace, I'd take Laplace any day of the week. And happily, the functions in the LME4 package, GLMER, that function will do Laplace by default. So will the GLMM function in the GLMM TMB package as well. You can see all these over here, LME4, GLMM TMB over here in the Laplace approximation row. If you're hell-bent on using PQL because you like your estimates underestimated or something, like your estimates rare, maybe, would that be the expression? Proc GLMMs, if you're a SAS fanatic, there's nothing wrong with SAS. I used SAS for a long time when I first started teaching at Griffith, probably for about 15, 16 years, actually, before we moved across to R. SAS is fine. So it's got Proc GLMMs, and there's obviously GLMM PQL in the maths library in R. For Laplace, we've talked about. Also a very interesting and useful package, and one that I'm not going to make any promises, so Brooke and Amy, if you're listening, I apologise for maybe getting people's hopes up here, but maybe at some point in the future, I wouldn't mind running one of these red workshops, or maybe a couple, on using R-INLA, which is integrated nested Laplace approximations, and it's a way to do Bayesian analyses using Laplace approximations. And the models that you can do with R-INLA include spatiotemporal. I had a student for a capstone just last trimester, actually, or T2 or T1 and 2, sorry, analysed the entire mainland COVID cases from 2000 to 2004 for every county in mainland US with a spatiotemporal random effects model. And we did that in R-INLA. So INLA is extremely flexible and interesting package that would help you do spatial, temporal and spatiotemporal modeling as well, which is really useful. There's the GLMER package and LME4 function in the LME4 package, GLMM TMB function in the package of the same name in R. if I had the choice, if I was in the situation and I had the choice, I would always choose, if I'm doing frequentist work with generalized linear mixed effects models, and by frequentist, I mean not Bayesian, adaptive Gauss-Hermite quadrature is the gold standard, in my opinion. And I know I'm probably a little bit biased, but it generally is, at the moment anyway, current gold standard. Unless you're a Bayesian, in which case, Markov chain Monte Carlo is probably your thing. I don't necessarily disagree with you, but most people don't want to sit around for 25 hours while their chain converges. So most people want something that pops out an answer within 10, 15 seconds, in which case it puts you squarely in the frequentist world. Unless you're doing some inler stuff, but I'm rabbiting on now, so I'll stop. Okay, the adaptive Gauss-Hermite quadrature. There's my package, which isn't available on CRAN, but if you email me if you want to try it, you can, and I'll send it to you. My package is GLMMNQ. There's GLMMML package of the same name, function of and package of the same name in R. There's the GLMER function in LME4 that will do adaptive Gauss-Hermite quadrature, but only on one random effect, only on a random intercept. Same with GLMMML. It'll do adaptive Gauss-Hermite quadrature, but it'll only do it on one single random intercept effect in your model. So you're limited there. If you need more than one random effect, you probably have to use Laplace in the previous row. There's proc NL mixed in SAS. That's been around a long time, NL mixed. Seems to do a pretty workmanlike job. I don't know Stata particularly well, but in the last workshop, I think there was a couple of Stata users, so I thought I'd throw in XT Logit from Stata. But don't ask me about that one. I haven't used Stata for probably 30 years, maybe. Okay, and then finally, the Monte Carlo, Markov chain Monte Carlo, whatever method. There's a few packages in R. There's a few standalone packages like WinBugs, JAGs. In R, you've got MCMC GLMM. There are plugins to give sampling packages and Stan in R as well that you can use if you want to go Bayesian. You might notice in the pros for this one, I've said a pro is that it's Bayesian, because essentially you scratch the surface on most people who studied a lot of statistics or who are statisticians like myself. You'll find they're probably Bayesians at heart, especially when it comes to p-values and things like that. And I consider Bayesian to be a pro generally. But you also might notice that I've put Bayesian in the cons column as well, because Bayesian approach to this stuff can also be a major pain in the butt and create headaches and complications that a lot of people don't necessarily care about trying to figure out. So, all right, postulating an initial plausible model. How do we go about when we're confronted with this potentially complicated sort of data? How do we go about figuring out what initial model might be plausible for us to fit to the software? The first thing you need to do is identify the response distribution. Okay, is it binary? Have you got presence absence data? Is it count data? Is it the number of things that are occurring per unit area or per unit time? Is it multinomial? Is it ordinal data, like our SET and SEC scores? Is it ordinal, like the Likert stuff that the university averages, despite me telling them that's such a bad idea? But okay, so all of these things. Think about those things. Identify what is the distribution, what's the conditional distribution of the response. That's step number one. Same step as step number one in GLMs as well. Then in doing that and looking at that variable, look for unusual is probably not the right word here, but look for variations around that theme. So for example, in +on data, in count data, it's not uncommon in certain academic areas like ecology and environment or other places as well for the count data to maybe have an inflation of 0 values for one reason or another. So is your data got more zeros than you might expect a Poisson to have, or is there a theoretical or mechanical reason why you would believe that there could be another process generating, or another process in combination with what's generating your counts to create more zeros than you might expect? Now, figuring out whether you've got zero inflation is usually something you do after you fit the model, but sometimes you can pick it that it might be something that needs to happen or be considered prior to running the models. Consider how the data was collected. So if you've collected your sites through space, so if you're in an environmental situation, a river situation, catchment or whatever, and you've collected information on sites, you're looking for the number of koala scats in Toohey Forest or something like that, in Transx or whatever. If you've collected your data in sites and those sites are geographically linked, then it may mean that you might need to consider spatial dependency in your data, for example. If you've collected things through time, over time, like the bacteria data where we collected nasopharyngeal swabs from Aboriginal infants at various moments over 11 weeks, then maybe you may have some spatial dependencies. Or if some of those infants were from the same family, there may be familial dependency that you need to think about, right? So think about these things. Identify the clustering structure. Is it a single level repeated measure like in the bacteria data set we looked at? Is it nested, hierarchical like in the SET or SEC example I gave you right at the beginning? Is it crossed, etc.? Visualize the response in some way. These are all just tips, right? These are general modeling tips, not just limited really to GLMMs, to be honest. Visualize the response and the covariate relationships if possible, and it makes sense to do so. Panel plots, which is a plot per group, is often useful. What they call spaghetti plots, where you have a line for each of the groups, and they sort of look like, some people say it looks like spaghetti. And if you can, for GLMMs in particular, or GLMs as well, see if you can't plot the response on the scale of the link function instead of on the raw response scale, so that you can actually see whether your explanatory variables are linear, have a linear relationship with that. If you just plot the raw counts from a Poisson random variable, for example, against numerical covariates, you probably expect to see some sort of logarithmic relationship if your GLM model is correct, because that's what it specifies in the link function. If you can convert it over to the link function scale before you do that graph on the Y variable, then you can look for linearity. People can see linearity better than they can see logarithmic relationships. Trust me. All right. When you postulate your initial plausible model, you then need to think about the fixed effect structure. Which covariates do you think have a population effect on the response? And potential correlations could be involved there. And then think about your random effect structure. Do you need just a random intercept? Will that be adequate? Are there other covariates that you think may be contributing to the random variation you're observing between the groups? If so, you may want to consider one or some of those as random slope terms in your random effect structure. If you consider having random effects, or more than one random effect, then you do need to actually consider as well the random effects variance covariance matrix structure. So do you want these random effects to be broadly estimated with no constraints, and therefore you want them to have their own specific correlations with each other? Are you happy to assume they're uncorrelated and just fit what's known as variance components, random effects, covariance structure, etc., etc.? These things all play into the complexity of the model and the numerics of what you're going to have to do when you fit it. With model fitting and selection, which estimation method should we use? Well, see that previous table. But here's some guidance as well, based around other things, not just on the previous table. If you have a relatively small number of random effects, e.g. random intercept, maybe random intercept and one random slope, use adaptive Gauss-Hermite quadrature if you can. But keep in mind that the current software that I've found in R to do Gauss-Hermite, it limits you to a random intercept per group only. Okay, so actually I probably should have changed that there to say if the number of random effects is 1, then you can use adaptive Gauss-Hermite quadrature, in R at least anyway. For larger problems with more complex random effect structures, Laplace is probably your best bet, or at a stretch, PQL, or if you really want to be a Bayesian about things, Markov chain Monte Carlo approaches, Bayesian approach. Go full Bayes. Responses with issues, for instance, if you discover that you've got zero inflation or over or under dispersion in your model, we'll have a look at that in an example we're about to get to in a minute. That's software dependent. So for instance, R-Inler is very flexible, but has a bit of a steep learning curve. GLMM TMB, which uses Laplace, it has functionality to do zip or zero inflated Poisson or zero inflated negative binomial models. It can also do hurdle models. If you don't know the difference between zero inflated models and hurdle models, zero inflated models are sort of a mixture between a distribution that has zeros and then the actual distribution of your GLM. All right, so it's a mixture of the two, whereas a hurdle assumes that we have two separate processes and you're either in one category or you're in the other. So there's a separate process generating the zeros. Once you get past that hurdle, then anything belonging to the other process must be, for example, a Poisson where you truncate it so that the Poisson can't have zeros anymore. So that's the main difference there. And in some cases, it makes sense to use one over the other. Again, if you're not sure what I'm talking about there and you think you've got an issue like that, you can always come and have a chat with me when I have the time. Okay, so if you've got those sorts of issues, then it depends. You need to be looking at software that can help you do that. In R, the most flexible that I've seen at the moment anyway, for the frequentist type of stuff is GLMM TMB. Responses with small means like binary responses or Poisson with small numbers of counts and or large random effects variances. Instead of what I've written here, I should have put anything except PQL. If you're in a situation where you've got responses with small means or random effects with large variance parameters, then don't use PQL. Use anything but. Okay, model selection depends on the method you use as well. So if you're using PQL, you're out of luck. With model selection, you might as well just toss a coin and hope, right? So if you have a lot of covariates or a random effect structure that's relatively complicated and you intend to try to reduce that model by looking at it and using model selection tools like AIC or likelihood ratio tests, don't use PQL because you won't be able to do that with PQL. So use Laplace or something like that. So Laplace will give you AIC, log likelihoods, DIC, BIC, etc., or BIC. That's good if you've got no other choice, which I've been telling you now. I don't really think you do have much other choice in R at the current time. But for this purpose, model selection, adaptive Gauss-Hermite quadrature is the gold standard. It's just unfortunate that it's very limited in its scope in the current implementations. And again, if you want to go full Bayes, you've got the gamut of inference available to you. You've got model averaging, you've got Bayesian information criterion, deviance information criterion. You've got all sorts of good stuff, credible intervals, etc. So a few warnings. In generalized linear mixed effects models, as opposed to linear mixed effects models, AIC and likelihood ratio testing in particular are asymptotic. So when we do a likelihood ratio test, you get a p-value based on a chi-square statistic. The distribution of the likelihood ratio test statistic being chi-square depends on, or gets better as sample size gets bigger. Parameter testing for variance components, there's a boundary effect, a known effect of estimates on the boundary when it comes to this sort of stuff. There's especially for variances, we have that boundary at 0. Variances can't be negative, so they're bounded below at 0. And so if your model's estimated, or if you're constraining one of your variance components to be on that boundary of 0, then you need to be careful, because it's a known problem with maximum likelihood, especially, that you'll get conservative estimates in that case. Degrees of freedom issues as well. So these GLMMs are a little bit complicated, and no one really has good answers for some of these things yet. So if you do have an issue and you come to me, ask me about it, please be prepared for me to sort of give you multiple options and very many caveats around my advice. Okay. But there's also potentially degrees of freedom issues as well with AIC. You're talking about the population level, or are you talking about individual level AIC, and what sort of degrees of freedom we need to allocate to calculating AIC and differences in the models. Because as you may remember, AIC has a penalisation term depending on degrees of freedom. So it's rather important to get the degrees of freedom right. All right, assumption checking. We're actually going pretty good. Look at me go. Look for over or under dispersion. Look for zero inflation if you suspect it, if it's pertinent in your particular application. Look for dependency in the residuals, spatial or temporal, or both. That's always data specific and how your data has been collected. Generally speaking, if you've collected things through time, there's always the potential for there to be temporal dependencies or time dependencies. If you've collected things through space, depending on the nature of what it is you're collecting and the sort of geographical boundary of the effect of what you might be looking at, there could be spatial dependencies. And of course, if you're collecting things through space and time, like the spread of COVID, for example, or other diseases, then you may be wanting to consider spatio-temporal dependencies, which is an entire another kettle of fish that we won't go into today. But just be aware that those sorts of models exist. I really like this current simulated residual diagnostics for generalized linear models and generalized linear mixed effects models in a package called DARMA. I'm going to call it DARMA. It's diagnostics for hierarchical, the A is the archical bit, regression models. That's the name it started out with. So it actually started out with the name DARM, but the author of the package learned that DARM meant intestines or something in Hindu, and so whacked an A on the end. So he wasn't wandering around saying that his package was intestines, I guess. I don't know. It's a good little package, and we will see this. We will use this in a tick if I shut up and get to the examples. There's another package that I quite like called Performance, which automates a lot of the residual diagnostics as well. But you do have to be a little bit careful with what Performance pumps out at you for some of these GLMM things. Take it with a grain of salt. I would trust the diagnostics from Dharma more than out of performance if push came to shove. And always remember that the generalized linear models and generalized linear mixed models, the residuals in these won't look normal unless you're approaching situations where central limit theorem kicks in and large sample stuff and things become more and more normally distributed at that point. But generally speaking, that doesn't happen. All right, interpreting your estimates before we move on to the examples. For fixed effects, again, just like linear mixed effects models, fixed effects are the population effect corresponding to the x variable on the scale of the link function. So if you want to be talking about the scale of the response, you need to apply the inverse link function. Happily, you don't really have to do that manually nowadays. There are some nice packages in R that will apply that for you. And again, we'll see that in an example. And I'll make the R code for that example available. I think we'll only have time for one example this afternoon. So I'll pick the one that's informative over the one that isn't. Excuse me for a minute. I think there's a jet flying over. So using predict or these other packages is a better way to go about it nowadays, I think, and maybe visualizing the results rather than a table of numbers is often useful as well. Obviously, the table of numbers works, but obviously many people's eyes glaze over when you present them with coefficient tables and immediately understand what you're talking about when you throw a graph or two at them. So just be mindful of your audience and who you're trying to convince of what, and apply an appropriate visualization to that situation. I won't tell you this story. I was just thinking of a situation once where I wanted it to look complicated because I just wanted them to do what I told them to do, and I didn't want them looking at things too much. I wasn't doing anything dodgy. It was just like, yeah, look, I know what I'm doing. So here's this really complicated looking output table C, and they all nodded dutifully and we got things done. But so there could be situations like that where sometimes going complicated is better or suits your purposes. But generally speaking, keeping things simple is much, much better. Okay, take people with you rather than confuse them. That's the name of the game, I would suggest, especially in science and especially in empirical sciences like statistics. Okay, so there's all these other nice packages. We're going to use GG effects in a minute to take a look at fixed effects and random effects estimates visually. For random effects, the predictions are the what we call the conditional group modes. You could consider them BLUPs. I don't like to call them BLUPs because I'm not convinced that they're linear or best in the statistical sense. But some people call the estimates of the random or the predictions of the random effects you get out of a GLM, some people call them BLUPs, but I just call them the conditional group modes or means, whatever you picked. You can use them together with the fixed effects to get a group by group response profile if you wish, or you can just look at them on their own to see which groups deviate the most from the population. Remember, the random effects interpretation is how each group deviates from the population effect. So if you're looking to find groups that deviate the most from the population, then pumping out the random effects estimates or predictions and then, choosing the ones that are most extreme can help you identify individuals in the population that are least like the population, for example. Okay, you also need to look at them on the scale of the link function by using the inverse link to map back. Okay, all right. All right, so now we're going to look at an example in the remaining 20 minutes or so we have. So I'm going to get R open in a minute and we go over and we're going to take a look at the owls. I was going to do the bacteria one as well, but I don't think we'll have time this afternoon. But happily, the owls is a very informative analysis and shows you a lot of techniques and things you can do in R because this data's got a few idiosyncrasies and things we need to account for. Okay, so the owls data set's fairly well known in this sort of part of the statistical literature. Essentially, it's the food begging behavior of nesting owl chicks, whether there's an increase in it when the father's feeding them, when the mother's feeding them, is it related to the number of chicks in the nest, etc., etc., etc. Okay, so that's the data. If you want references, they're at the end for this stuff. But the Menzies School of Health Research annual report, that's for the bacteria data that we won't be analysing. Okay, and you can find that in Venables and Ripley if you wish in the maths library. Okay, let's go over to the desktop and where are we? Let's get R open. Now here's a script that I installed previously or created previously, and I will make this R script available to you at the end of the workshop so you can have a play with this yourself. All right, so first thing with R is we need to either install if they're not already installed or load the libraries if you've got them installed that we need. And don't worry, if you don't have these libraries installed in R, when you open this script file in R, it will say, RStudio will say, if you're using RStudio, RStudio will say, you don't have these libraries installed, would you like to install them? And you just click on yes, and it will install everything you need. Okay? All right, so the first thing is let's take a look at the data by viewing it. So we've got a nest ID. So what we've got here is repeated measures on nests, on owl nests, barn owl nests. Okay, so we've got a nest ID variable that we use to keep track of which group we're talking about. We've got a food treatment regime, whether that particular nest at that particular moment was in a food deprived state or a food satiated state. Okay, had they have enough to eat or were they starving essentially? The sex of the feeding parent, male or female. Arrival time, we're not going to use that. It's not important, but that's how long it took for them to arrive with food. Sibling negotiation is our main response variable. It's the number of begging attempts by the siblings in a nest in a certain time period. So for instance, there were 4 attempts at begging from the male parent for food in this particular nest when it was in a deprived food state in that top row. Brood size is how many chicks are in the nest. And we're going to use that as what we call an offset in this model. It's got a known effect. We know it has an impact on the number of begging attempts, i.e. the more chicks there are in a nest, the less food there is to go around, therefore the more begging will occur. So we know it has an effect, but we don't care about its effect. We're just going to put it in as an offset. So when you have a variable in your data that you know impacts the response variable of interest, but you don't actually care about estimating it because you're essentially aware of its effect, you can add it in as an offset. Okay. All right. Or, the alternative would be to divide your response variable by brood size, but then that would create a situation where we don't have counts for the response anymore, which makes analysis potentially more complicated than just doing a +on GLMM. Okay, so I've given the game away here, but because we've got counts of the number of begging attempts in a specific time period, I'm going to postulate that this data has a +on distribution, and we're going to fit a +on generalized linear mixed effects model to it, initially anyway, and then see what there is to see by running diagnostics on our model and updating it depending on what those diagnostics tell us. All right, so the first thing we should always try to do if possible, and this is a pretty half-hearted attempt, at visualization on my behalf, and I do apologize. But we should try to visualize the response and the major covariates. So here I've done the number of begging attempts, sibling negotiation on the y-axis by deprived or satiated state. That's why there's two columns of numbers in the picture, per nest. So this is a panel plot per nest. And I can do another one for the sex of the parent as well. See if there's anything to see. It's hard to see anything in these pictures, to be fair. I'll probably do some more interesting things than what I've done, but time was limited. So I made a stab at visualization, but we're going to jump straight into the analytical approach. Okay, so we're going to try a Poisson GLMM with a single random effect per nest, so a random intercept per nest, just to say that these nests differ from each other in their begging attempt numbers and the number of begging attempts that occur within the nest over repeated measures is probably going to be correlated or dependent. So adding that random effect, that single random intercept random effect in there is going to at least help us on the journey for this modeling process. Okay, so fit1.L, I'm using GLMM TMB here, which is going to use Laplace approximation. And this is the code. I won't go through the code with you. I'll just show you essentially what it outputs. I'm also going to use GLMER as well, just to say that you've got that code. When I give you this script file, you'll have the code to do it in various R packages. GLMER, again, if you just say the number of adaptive Gaussian quadrature points is 1, that's equivalent to, and I think that's the default actually in GLMER, that's equivalent to Laplace. So fit1.L and fit1.QL here, they should give us roughly identical results if the algorithm's been implemented properly. And I've also done GLMER, but this time with 10 quadrature points. So this one is the adaptive Gauss-Hermite quadrature with 10 points. So Q2 model is the one where we're using adaptive Gauss-Hermite quadrature, and the previous two we're using Laplace. If you take a look at the outputs between the three, you'll notice that the estimates, and I'll let you do this in your own time, but you'll notice that the estimates are very similar, but there's a major difference here with the Gauss-Hermite quadrature. You'll notice that the AIC and the BIC, for example, are around 3,500. For the two Laplaces, it's more like 5,200. So I would trust the Gauss-Hermite quadrature here more than I'd trust Laplace for this stuff. But on the other hand, the absolute values, the magnitudes of AIC don't matter. It's the relative size of the AIC between two models that matter. All right. So I wouldn't be too concerned about that. It's the parameter estimates being similar that we care about. All right, so there's very few differences. Now, I'm going to use the TMB Laplace, GLMM TMB, in what remains of this example, because it turns out there's going to be a few things going on here, and the TMB function for GLMMs is much more flexible than GLMER. Okay, so we're going to, we fit our model. We probably shouldn't really have taken a peek at the estimates at this point. What we really should do after we fit a model is check the assumptions using diagnostics, model diagnostics. So this is the performance library. It's going to create some automatic diagnostics for us eventually. It's a bit slow. Eventually, come on now. There you go. All right, so some of these are useful, some are not. So this one in the top left corner is useful. You see the green is the observed data and the blue is the model predicted data. And if the model is predicting the data accurately, then the two colors should overlap and coincide. What you can see here is that the model predicts a certain peak in the counts down here. that's not matched by the actual data. And in addition, the model is not predicting the number of zero counts that is in the data, right? So we've got over 150 zero counts in the observed data, and the model's not picking that up, okay? Because the model is assuming that the distribution is Poisson, and while the Poisson can account for some zeros, it can't account for excessive zeros. And so this picture is telling me that we've got an issue here with, well, a couple of things, but zero inflation is probably one of them. The other thing I'm looking at, I don't really look at this top right picture much. The quantile residuals look particularly poor in the bottom left, and the normality of the random effects seems fine. Okay, but if I really want to understand what's going on in a little bit more detail, I can use the simulated residuals from the Dharma package. We don't have time to really talk about the Dharma package in detail, but essentially it simulates the residuals from the GLMM so that you can compare them to a uniform random variable between 0 and 1. And so you can, instead of sort of looking at a QQ plot for a normal, you're looking at a QQ plot, if you like, for a uniform 0, 1, excuse me. Okay, so first we do our simulated residuals, then we plot them. Now this creates two generic plots. Okay, the first one is a observed versus expected if you have uniformly distributed simulated residuals. Pretty clear here that we don't. We also see in the bottom left of this picture, if you look, that there are far more observed residuals down near 0 than you would expect if it was really Poisson distributed. Okay, so you've got this sort of, well, S shape here, which is an indication that we've got 0 inflation. But also, I think we've over dispersion as well. Maybe because of the 0 inflation, but we need to think about over dispersion here as well. Remember I said in the theoretical part that for the Poisson and the binomial, the dispersion parameter is mathematically fixed to be 1. Well, when there's more dispersion in the data than fixing that dispersion parameter to 1 can account for, that's known as over dispersion. Okay. If we could set the dispersion to 10, which we can't, but if we could, then, you know, maybe that would account for it. But we've got over dispersion here, and I think we've got 0 inflation as well. This picture on the right, what you're looking for in this picture is basically, this is plotting model predictions, rank transform, so that puts it on the 0, 1 scale on the x-axis. You've got these simulated residuals on the y, and you've got quantile regressions in the dotted black lines. Okay, and what you're hoping to see versus the actual, so the dotted black lines are the theoretical quantile regressions if the residuals are okay. And the darker or the red lines with the grey around them with the confidence intervals are what you're observing. Okay, so what you're hoping is that these things line up on the dotted lines. If they don't, you've got some issues. Okay, and it actually tells you. It also puts a few hypothesis tests on the graph as well. So the Kolmogorov-Smirnov test to see if these residuals actually follow a uniform 0, 1 distribution is this one. P-value is 0, so there's significant deviation from that. There's a dispersion test. It's just non-significant, but I believe there's over dispersion problems in this data, so we're going to deal with that. And then there's an outlier test. Given that the residuals don't align with what we're expecting, don't pay much attention to the outlier test at the moment, because it's just picking up the fact that we've got a ton of zeros down the bottom. So we need to fit another model accounting for these things. Okay, so we've got some issues. We've got dispersion issues. So that's the one we'll deal with first. We'll assume we're going to treat the dispersion. It's common practice when you've got over dispersed plus on or count data to try what's known as a negative binomial model. In GLMM TMB, there are different versions of the negative binomial that correspond to different parameterizations of how the dispersion parameter is allowed to vary with the mean. So in the negative binomial, the dispersion varies. It's got a variance function, essentially, like that varies with the mean. So there's a quadratic parameterization, there's a linear parameterization. We're just going to use the linear parameterization for the negative binomial dispersion here because it's simple and you can directly relate the over dispersion to the value of the estimated dispersion parameter when you do linear. When you do quadratic, it's inverse relationship. So the smaller the parameter estimate, the more the dispersion. So we're just using, in this case, n binomial 1 for the family, which is the linear parameterization for negative binomial. We're going to check these diagnostics, see if it looks better. We're waiting. It takes a while. Okay, now you can see top left now, the model's doing a lot better against the observed data. The dispersion, accounting for the over dispersion has helped somewhat, but I still maintain there's a little bit of zero inflation going on here. The quantile residuals down the bottom left look a lot better. Okay, so we've solved quite a few problems just by allowing over dispersion here. We can also check the Dharma simulated residuals. Okay, there they are. That looks a lot better. And we can test the dispersion as well. Now, you may say that we've gone too far and the over dispersion, now we might have under dispersion, but when the dispersion's flicking around like this, it's generally an indication something else is going on. Usually that something else is zero inflation when it's count data. So now, on top of our over dispersion model, we're going to add zero inflation into the model, and we are going to allow that zero inflation to depend on the food treatment and the gender of the parents as well. Okay, so the probability of seeing more noughts than we might expect to see. Thanks, Erwin. I'm sorry I'm running late. Sorry about that, but thank you for being here, mate. I appreciate it. Okay, so we're adding over dispersion and zero inflation now into our model. All right, that's run. We're going to check that diagnostic or that set of diagnostics. So you see the pattern here. We fit a model, we look at the diagnostics, we use those diagnostic graphs to help us diagnose what the problems with the model are. Once we figure out what those problems might be, we refit a new model to help combat those problems, and we run the process again. We check the diagnostics of that new model and so on. And it's an iterative process. This is the modeling process. Postulate a model, fit it to the data, check its assumptions with diagnostic tools, modify the model depending on what you see going on in those diagnostics, rinse and repeat until you get to a point where you're happy with the model fit via the diagnostic process. So you can see here now, top left picture, we've got excellent agreement between the predicted model data and the observed data. Very, very good. So having the over dispersion in the model and accounting for the zero inflation in the model has really helped there. Our quantile residual distribution bottom left looks particularly good as well. We can check out the simulated residuals. Again, you can see the expected versus observed here on the left is looking excellent. We've got no problems here with the right either. Kolmogorov-Smirnov p-value is very non-significant. So is the dispersion, and so is the outlier test. So I think we've probably hit a winner here with this particular model. Okay. Now, once we've got this, we can do some other pictures as well. Just quickly show you. Hopefully you guys can stay another couple of minutes. We're almost done. Okay, so we can plot the residuals for the variables as well in the fixed effects variables. So our deprived satiated food state, and you can see that everything looks very uniformly distributed there, no problems. We can do a dispersion test. It's low, but it's fine. And we can do a zero inflation test as well. So the red line, the grey data is simulated, the simulated values you might expect to see based on the data in the model. So it's almost like bootstrapping. It's permutations and simulations, right? And the red line is where your model actually sits in those. And so if it's sitting toward the middle or away from the extremes, that's not a bad thing. Okay. So I'm going to skip a few bits here that you can have a play with yourself and go down to now. I think we've found the model we want. Have we? I think so. We found the model we want. Let's use it to visualize our results. Okay, so the first thing we're going to do is use the GG predict function to visualize the fixed effects. All right, so this is the fixed effects taking into account the random effects, but not taking into account the zero inflation. So this is just over the mixture model that the zero inflated model assumes. And you can see that while there doesn't appear to be a gender effect in the predicted counts of begging, all right, so the blue is male, red is female. There's no real difference, and the bars are the confidence intervals around them. There's no real difference there, but you can definitely see in either situation, but you can definitely see that there's a difference between the food deprived and the satiated numbers, average numbers of begging attempts. And it passes the common sense test, right? If you asked yourself, how many begging attempts would you expect to see in a nest of starving chicks compared to a nest of fully happy and well-fed chicks? Everyone would say, well, you'd expect to see more begging effects in food-deprived nests than in satiated nests, and we're seeing that here. So always use your common sense when you're looking at these results too, to make sure you haven't screwed anything up in the model, put something in the wrong way. It happens. Okay, so always do that. Now, if we want to tease out what's going on in just the Poisson part, in just the non-inflated zero part, we can actually ask GG predict to do that as well. Okay, and you get even more stark. Look at that. So what this is doing is this is filtering out those extra zeros, getting rid of them, and just conditioning on the other part of the model, the part of the zero inflated model that says, okay, this set of observations are from the count bit, the bit that's producing the count, the process that's producing the counts, and we're going to just filter away the other process that was generating the extra noughts, the extra zeros. Okay, so when you do that, it all looks even more interesting. And in fact, maybe at this point, you could consider putting a gender of parent and food treatment interaction in there if you wanted to, just to see. We can do the same thing for the random effects. So that was the fixed effects. We can do the same thing for the random effects if we care about the random effect conditional modes. Again, this is across the entire observations, not differentiating the extra zero process from the zero and count process. This is just across the whole thing. These are the random effects. If you filter out the process that's generating the zeros on top of the Poisson counts and look, you get a much more spread out situation. Okay, and you can see which are extreme nests. So for example, these guys up here, I can't really see the names of the nests down the bottom, but these ones up here you might consider to be nests that have an extremely large number of average begging attempts from their chicks. And if you were, I don't know, doing wildlife conservation for barn owls, you might want to focus in on what was similar about these particular nests, if anything, to see what might be going on, because clearly the chicks are hungry. Maybe geographically these nests were all in the same area. It'd be worth checking out anyway. So these sort of random effects can point you in the direction of places to look for further study and research. Okay, and then finally, we can actually look at the zero inflation probabilities. And the variables that contribute to it. So you see on the y-axis here are the zero inflation probabilities. So these are actually the probabilities of not being in the extra zero group. How do I explain this? Just let me very quickly. So you've basically got a mixture of models. You've got a probability pi of belonging to a group that generates the extra zeros. And then you've got 1 minus pi probability of being in the group that does the Poisson, the counts. Okay, this picture is showing you how the explanatory variables, the sex of the parent and whether it's deprived or satiated food regime, predict being in this Poisson count category. All right. Where are we going? Desktop. All right. So you can see that in the deprived, for the deprived food treatment category, you're not very likely to be, you're highly likely to be in the extra zero. Right. There's only a roughly 10% chance you're in the actual counts for begging. The clearly being in a deprived nest state, food deprived nest state, is what's primarily generating the extra zeros. Okay, for some reason. Okay, and sorry. Yeah, that's right. And then in the satiated state, you've got a much higher probability of just being up in the normal Poisson counts. Okay, I might have screwed that up actually. My brain's gone to mush, but I can't use it to remember at the moment. I'm sorry about that. But if I've made a mistake, I'll let you know. But I think I'm right. Okay, that's it, guys. Thank you so much for hanging in there with me. I'll pop that script file in to chat now so that you can have a play with the owls yourself. I've extensively commented it so that hopefully you'll know what it is talking about. How do I... What's going on? Come on. Sorry, now I'm having fun with, there we go. Having fun with Teams. No, I don't want to add a new app. Why is it telling me about adding apps? It's been a long afternoon. Seems like I can't. Look, I'll make sure that I get that script uploaded. I might have to play around with it. I need to let you guys go because I've already, again, kept you longer than the 2 hours. I do want to thank you all for hanging in there with me. I know it's probably been very full on. I hope you got something out of it this afternoon. And again, open invitation to get in touch if you need advice or help. Okay, enjoy your afternoon, guys. Talk to you later. Very welcome, everyone. Very welcome indeed.